We have Tobias Ensslen from Hanchikat, and I'm very excited about this talk. So far we heard a lot about, and also in our previous meetings about pore-forming toxins and bilayers and so on, but now we have really on live cells. And this is very exciting. I had a privilege to see a couple of your slides in advance. They were really, really awesome. And I hope everybody else likes them as well. Tobias, floor is yours.
Yeah, thank you very much for the introduction. Now the expectations are quite high, I guess. So first of all, thank Nanion for bringing up this super cool meeting here every year. It's always a pleasure to be here, and especially Sonia for inviting me here and giving me the chance to share with you very preliminary, very recent data we acquired with the AtlaZ system here. For those of you who do not know me, as already mentioned, I'm a nanopore guy. So I'm coming from the field of nanopores, and we already had talks yesterday, but just to remind you what nanopores are and what they really do. So nanopores are protein channels in membranes, and what people do is they apply a voltage gradient across these membranes, and then they watch molecules traverse or interact with these nanopores, and they can measure these funny signals. They call it resistive pulses. So in the end, nanopores are nowadays used widely as single molecule sensors with a very high resolution. However, this is not what nanopores are designed for. So nanopores, actually, they also go with the name of pore-forming toxins, and they are produced by bacteria. And these things do really nasty stuff. So they can cause actually severe algal gangrena or necrosis. And depending on basically the type of nanopore you got an infection with, the mortality rate is extremely high and can reach up to 100%. So there is a certain interest of understanding how these nanopores work and what they really do to the cells. And in last year's meeting, I already gave a talk about an approach towards this when we used the synchropatch device. And in this little endeavor here, we patched single cells and we applied nanopores here, and we watched actually the cells to die from our nanopores live. And to make this long story short, what you can see here, is there a laser working? Okay, what you can see here is basically the main outcome. So we were comparing Aerolysin, which is a pore-forming toxin coming from an Aeromonas bacteria, and this is very efficient. So this killed almost every cell immediately. Whereas Sarol, this is another PFT, it comes from a different organism, but its sequence is very homolog to Aerolysin, showed a completely different behavior. And what we found here was a high specificity for cells which had a certain molecule presence or a receptor present on the surface. And just to give you an idea what this receptor is, this is called GB3. This is a globosyl ceramide. So this is a tri-sugar. And interesting enough, this sugar has been shown that it is associated to different cancer types. So in cancer cells, this is overexpressed, and it's found, for instance, on lung and colorectal cancer cell lines. But for me as a scientist, this is a super interesting molecule because we can do a lot of science with it, because it gives us a very high level of control in our experiments. So for instance, there's another molecule which is called PPMP, or abbreviated PPMP, that has some structural similarities to the GB3. And when you apply this to your cell culture, you can actually basically inhibit the synthesis of GB3. So you can generate tumor cells which do not have this receptor present on their surface. And in addition to that, there is another molecule called PNPG, and this is actually a molecule that can act as a soluble GB3 competitor. So this system basically gives you the entire control in your experiments of having positive, negative, and competitive cells here. And with this system in hand, we ask ourselves whether we could use impedance spectroscopy to really monitor pore-forming toxins acting on cells. So our procedure was actually quite easy and straightforward. So what we did here, we did regular cell culture, and then we plated the cells, or we seeded them in the 96 well format of the AtlaZ system. So you can see here, each of these 96 wells is housing a single tiny microelectrode. This is a working electrode, and we have a larger reference electrode around. And the entire machine was then placed inside the incubator, and we watched our cell signal to reach a plateau level. And the moment we reached a plateau level, we basically added manually our pore-forming toxins to each well, and then we watched the cells suffer from our nanopores. And basically this is what you get. This was the first result. So here I show you a graph which shows you this sorrel pore-forming toxin acting on GB3 positive H1299 cells. So this is a lung cancer cell line. And what you can see here is actually, as expected, it is a decrease in our cell signal, and it's a decrease in a concentration-dependent manner, actually. So this was already super interesting for me because it was exactly what we were looking for. But as always, when you look close enough, you find further stuff, which is a bit interesting. So the decrease was expected. This was cool. But especially here in the beginning, we always saw these sort of bumpy signals. So there was a positive bump, and we said, well, what is going on there? And we thought this could maybe be actually a swelling of the cells, osmotic swelling. So we did an optical investigation of our cells, and indeed, after 5 hours, we could see that our cells had an increased size of about 27%. We did the same experiment on GB3 depleted cells, so GB3 negative. We saw the pore-forming toxin induced effect actually to be reduced by a factor of 100, which was cool, but still we expected something like 0. So what is going on here? So maybe we thought our GB3 depletion was not successful, but we could verify this by the use of toxin, which is a highly specific GB3 binder. We labeled it optically. With a dye here, and then you can see that it binds to our GB3 positive cells, but on the depleted cells, there was absolutely no binding. So GB3 is not there anymore, but apparently there are other receptor molecules which can be recognized by sorrel here. Yeah, precisely. So when we changed the cell line, so we moved from lung cancer cells to colorectal cells, so HT-29 cells, they are also reported to be GB3 positive. We were very surprised with the result we found here because we did not see any action from or by sorrel on these cell lines. Maybe for super high concentrations, we saw a prolonged swelling phase, but actually there was no real insertion going on here. And this is something very surprising. We are still trying to figure out what is going on here. Excuse me, but the bottom line is actually that sorrel acts on different cell lines really differently. And yeah, for sure, we also check whether we have indeed GB3 on these cells, and yes, they are positive, but sorrel doesn't recognize it. So for the next experiment, we were really intrigued by this osmotic swelling, and we said maybe we can come up with an idea of control this osmotic swelling. And then we digged into literature, and we found an old paper published in the mid-80s where they used dextrane as an osmotic protection agent. So we said this is actually quite nice because when we would have dextrane present in our medium, the pores should still insert into the cells, but the dextrane should prevent actually the osmotic swelling. But the moment we wash it out and replace the medium by a fresh medium, we could maybe cause the cells to swell on purpose. So this is our data here. As you can see, we had an initial lag phase here, but after the lag phase here, cells, or actually wells, experiments with no dextrane, we saw again the expected swelling, so pretty cool. But all these experiments which had dextrane present in the media, they did not show the swelling, but the perforation, so the insertion of these sorrel pore-forming toxins into the membrane. And the moment we started to wash out, as expected, we could really see a very strong swelling behavior of the cells which were just perforating in the beginning. So this experiment was a really huge success for us, and it proves somehow that we have here indeed an osmotic swelling behavior. Negative controls for sure were done to show that dextrane doesn't have an effect on the cells itself. And what we then did next is we already changed the cell type, but now we changed the PFT type. And when we compare the sorrel with the super potent Aerolysin, you can see also here that Aerolysin kills either H1299 cells as well as HT-29 cells very efficiently, very fast, and we barely see any concentration dependency here. But it also causes a strong osmotic swelling artifact in the beginning of the traces. This here is my last technical slide. But I'm very, very excited to share this with you because I tried here to compare now different methods we had used in this system. And I did it in a way that we calculated also the area under the curve, and we made these nice dose response curves here. And I would like to highlight especially the red one, because this is the Sarol protein acting on H1299 GB3 positive cells. And when we compare this with published stuff, so this here is a cytotoxicity assay they did on the same cell lines, but with classical methods, we saw, or they saw actually the onset of the effect taking place at 68 nanomolar, and this is precisely where our red curve starts to grow as well. So we got a pretty cool correlation here. And even more striking when I compare the EC50 value we got here in the AtlaZ system from a very first, this is a single data set of 30 wells in total. We got a EC50 of 1.2 micromolar, and with the synchro batch 1 year ago, we found 2.3 micromolar. So this is actually pretty close to each other, and I was very happy to see that. So this means in the end, both devices work actually very great, very good, and they are comparable to classical standard methods, which are already published out there. Of course, we also looked at the Aerolysin, but as expected, it's already saturated here. This is also something which was reported a couple of years ago, that Aerolysin is actually active at sub-nanomolar concentrations because it's this super potent Aeromonas thing. And yeah, with this, I'm already at the end of my talk. I hope that I could convince you that the AtlaZ is a real powerful system that can give you a lot of data. So data analysis is still going on. The EC50 values, I just analyzed them last Friday, so this is brand new data. And I hope that I will be able to deduct a bit more, more model stuff here. And with this, I would like to thank you for your attention. I especially would like to thank Anna Sophia. She's a PhD student in Winfried Römer's group, and she helped me with all the cell culture for this experiment, and she also did the entire experimental phase together with me. Special thanks to Martin and Sonia for their great support here. Whenever we had questions about these systems, they were there, like 24-hour service. This was amazing. Thank you so much. And lastly, I would like to mention my own group. So I started like 2 years ago in own group, and in addition to nanopore measurements, we also offer recombinant protein synthesis, peptide synthesis, and mass spec as a service technology. And we are very open for collaborations. And yeah, with this, I would like to leave the floor for questions. Thank you.
Thank you very much, Tobias. Any questions? Yes,
Very nice, Toby. Maybe this is the obvious question, but the one concentration of Sarol where it sort of starts to recover again, do you know what's going on there?
Oh yeah, on the long hand side. No, we have no clue. This is actually something we are also investigating at the moment. This is something we saw throughout all the experiments. If we wait long enough, they start to recover, which is surprising because we killed almost all the cells. But maybe, and this has something to do with the plateau phase. It is supposed that when you reach this plateau phase that you have a monolayer of confluent cells. But we tried to monitor these cell layers, so with the transparent plates you offer, and we saw that we not really have monolayers. It's more like a multi-layer system. So my idea would be that when we add the pore-forming toxins, they are maybe just killing the surfacial cell layers, and when we have maybe an intact layer surviving down there, this might then start proliferation again.
So I wonder whether the swelling pace depends on the presence of the extracellular calcium, or maybe do you know what is the mechanism of this swelling?
Oh, this is a very good question. I don't know what the reason for it is actually. So actually also when you go to literature, it's not very clear. People claim whenever there is pore-forming toxin as a topic that they kill cells due to osmotic swelling, but nobody has really looked into detail where this is coming from. We were actually now happy that we could see it for the first time, but this is for sure ongoing research we do. Thank you.